Students, hope you all are doing fine. This is the question paper of MSc Microbiology Entrance Exam, and I am Dr. Tripti Malik Ahuja. This video is important for the students who are preparing for the different entrance exam for admission to the MSc Microbiology, Biotechnology, Biochemistry, Biological Sciences, Life Sciences, Bioinformatics courses. It is important for the students who are appearing for the common entrance test for various PG exams. In the playlist, you will find number of the videos which are specifically for different entrance exams. These are based on the multiple choice questions. If you are new to the channel, then do subscribe it and press the bell icon so that you can get the notifications of the new upcoming videos. In the description box, you will find the links of all the videos also. And one more thing, in the description box, you will find the links of some books which will help you in the preparation of the entrance exams. They contain multiple choice questions for different biological subjects. So now let's get started. The question number 1. Before I start reading the questions, I request you to take a pen and paper with you so that when I am reading the question and then the options, you can guess which one is the correct option. You can write the option on the paper, and I am going to tell you the correct option also, so you can match your answer with that of mine. If there is any doubt, you require an explanation, then you can write in the comment section. I will reply to each and every comment. So the first question is, which of the following doesn't have a cell wall? The options are mycoplasma, cyanobacteria, archaebacteria, and none. So out of this, the correct option, I hope you have guessed it correctly. It is mycoplasma, also called PPLO, which stands for pleuropneumonia-like organism. They lack a cell wall. Here goes question number 2. The causative organism of typhoid fever is a very simple question, and the options are Clostridium perfringens, Vibrio cholerae, Salmonella typhi, and Salmonella species. I hope you have guessed it correctly, and the correct one is Salmonella typhi. Salmonella typhi causes typhoid fever. Salmonella paratyphi causes paratyphoid fever. Together they are called enteric fever. Question number 3. Anthrax is a disease caused by option 1 gram-positive bacteria, 2 gram-negative bacteria, 3 both, 4 none of the above. So again a simple one. So this answer is the gram-positive bacteria. Anthrax is caused by the gram-positive rod-shaped bacteria known as Bacillus anthracis. Question number 4. The atmosphere at the time of origin of Earth was 1 reducing, 2 oxidizing, 3 mixed, 4 without gravitation. So the correct one is reducing. When there was the time of origin of the earth, then there was no oxygen. The gases which are prominent were the nitrogen and carbon dioxide. Question number 5th. Find the correct statement about photosystem 1 and photosystem 2. So this question is related to photosynthesis. The options are, the option 1, PS1 has 680 and PS2 has P700 as reaction center. Option 2, PS1 has 700 and PS2 has 680 as reaction center. Option 3, both PS1 and PS2 have P700 and P680 reaction center in alternate manner. 4th, none of the above. So out of this, the correct one is the second option, which says PS1 has 700 and PS2 has 680s reaction center. The first stage of photosynthesis is completed in the PS2, the second stage in the PS1. The 680 and 700, these are the wavelengths at which there is the maximum absorption of the light. Question number 6. Alcohol dehydrogenase enzyme belongs to the class. The number 1 transferase, number 2 ligase, number 3 oxidoreductase, number 4 hydrolases. So here the correct one is the oxidoreductases. Dehydrogenase enzymes for the alcohol are the enzymes for metabolizing ethanol in the body. Question number 7 says, which of the following processes doesn't occur in mitochondria? Note down, you should read it carefully, doesn't occur. Sometimes the student misses this doesn't or not term. Then they opt for the wrong option, so you should be careful while reading the question. The options are 1 protein synthesis, 2 DNA synthesis, 3 beta oxidation of fatty acid, 4 fatty acid synthesis. So the correct one is the first option, protein synthesis. Protein synthesis is never carried out in the mitochondria. Question number 8. Enzymatic reactions are characterized by lowering of 1 potential energy, 2 chemical energy, 3 activation energy, and 4 metabolic energy. So this was a very simple question, and the option, I know you have guessed it correctly, it is activation energy. Question number 9. Cryon's anatomy is a characteristic feature of 1 C4 plant, 2 camp plant, 3 C3 plant, 4 water submerged plant. So the correct one is the option 1, which is the C4 plant. Cryon's anatomy is seen in the C4 plants where tissue equivalent to spongy mesophyll is clustered in a ring around the leaf veins outside the bundle sheath. Question number 10. T4 cells determined by CD4 receptor are the option 1 MHC1 restricted, option 2 MHC2 restricted, option 3 helper B cells to produce antibody, option 4 not determined. So out of these, the correct one is the option number 2. These are MHC2 restricted. These are also called T helper cells. Question number 11. Which of the following enzymes require no template for synthesis? The option DNA polymerase, Taq DNA polymerase, RNA polymerase, and terminal transferase. So the correct one is the option number 3, which is the RNA polymerase. RNA polymerase causes the formation of messenger RNA from DNA, that means the transcription process. Question number 12. Sigma factors help to 1. RNA polymerase recognize messenger RNA in prokaryotes. Option 2 says RNA polymerase recognize promoters in prokaryote. Option 3. Small ribosome unit recognize messenger RNA. Option 4. DNA repair enzyme recognize breaks in DNA. So here the correct one is the option number 2, which is RNA polymerase recognize promoters in prokaryotes. Sigma factors are subunits of bacterial RNA polymerase enzyme. Question number 13. The enzyme which is used to add phosphate group to the 5' hydroxyl end of DNA is called 1. Phosphatase, 2. Dehydrogenase, 3. Esterase, 4. Polynucleotide kinase. So here the correct one is the option 4, polynucleotide kinase. It is also called PNK. I hope you are enjoying these questions and you're writing the correct options along with me. The question number 14, a very simple one. The predominant auxin in plants is, the option 1 is indole 3 propionic acid, option 2 indole 3 pyruvic acid, option 3 IBA, option 4 is IAA. So the answer is the option 4, which is the IAA. This is the most prominent auxin called indole acetic acid. It is a regulatory plant hormone required for the growth and development of plants. Question number 15. In ELISA, enzyme which is used for detection and visualization is 1. Conjugated with secondary antibody. 2. Conjugated with primary antibody. 3. Unconjugated with secondary antibody. 4. Conjugated with antigen. The correct is option 1, conjugated with secondary antibody. The enzymes most commonly used are the HRP, horseradish peroxidase, AP, alkaline phosphatase. These enzymes are conjugated with the secondary antibody and added in the second last step of the ELISA. It is conjugated the secondary antibody so that the same conjugate can be used for detection of various antigens. The question number 16. The major difference between northern and southern blotting is, the option 1. In northern blotting, gel fractionated RNA fragments are detected, while in southern blotting, gel fractionated DNA fragments are detected. Second, in northern blotting, gel fractionated RNA fragments are detected, while in southern blotting, gel unfractionated DNA fragments are detected. The option 3 says, in northern blotting, DNA molecules are used as targets, while in southern blotting, both DNA and RNA are used as targets. In northern, represented DNA probes are used for detection, while in southern, DNA RNA hybrids are used as probes. So out of this, the correct one is the first option. Northern blotting is used for the detection of the gel fractionated RNA, while southern is used for the detection of the gel fractionated DNA fragments. The question number 17. Which of the following sequences can be recognition site of a typical type 2 restriction enzyme? The options are CGG-CAA, option 2 CGCCGC, option 3 CTGGCC, option 4 GTCGAC. So as I have read the options, I request you to write these sequences in both the directions, the 5' to 3', and in the reverse direction 3' to 5', all the sequences. So as you will write these sequences, I am giving just a single pause, or you can stop the video, you can give a pause to the video and reconnect and then play it again. You can see that when we write these sequences, then out of this there is one sequence which when we write in either direction, whether 5' to 3' or 3' to 5', that sequence is remaining same. So that one is the second, CGCCGC. and when we write in back direction it is CG, CC, GC. So it remains same. Such sequences are called palindromes. Palindromes are usually 4 to 8 base pairs in length, and when they are read in either direction they are same. They are nucleotides, sequence of the nucleotides basis, they are same, and these are the recognition site for a typical type 2 restriction enzyme. So here the correct option is the option number 2. Question number 18 says, zinc helix and helix turn helix proteins are examples of 1. Proteins involved in protein targeting, 2. RNA polymerase subunit proteins, 3. Metal binding proteins, 4. DNA binding proteins. So here the correct option is the option number 4, which are the DNA binding proteins. These proteins, these are the structural motifs which are capable of binding DNA. The next question number 19. The enzyme required for first strand cDNA synthesis is, the options are DNA polymerase, option 2 E. coli DNA polymerase, option 3 Planoff fragment of E. coli DNA polymerase, option 4 reverse transcriptase. So out of this the correct option is, I think you have guessed it correctly. Yes, it is the option number 4, reverse transcriptase. Reverse transcriptase carry out the synthesis of DNA from the RNA, and this is the reverse of transcription. It doesn't follow the central dogma of the molecular biology. Question number 20. What is a virion? The option number 1, inactivated virus, option 2 viral capsid, option 3 ineffective virus, option 4 defective virus. The correct one is the 1, inactivated virus. Actually it is the viral particle which is present outside the host body. It is capable of causing the infection once it enters the host cell. The question number 21 says, lysozyme breaks the following linkage between bacterial cell wall. The option 1, N-acetyl murmuric acid and L-alanine. Option 2, N-acetyl murmuric acid and N-acetyl glucosamine. Option 3, N-acetyl murmuric acid and D-alanine. Option 4, N-acetyl murmuric acid and first amino acid in the glycine chain. Here the correct option is the option number 2. Lysozyme causes the hydrolysis of beta 1 to 4 linkage between the NAM is N-acetyl murmuric acid, and NAG is the N-acetyl glucosamine. Question number 22. Which of the following is not a neuroparasite? Not a neuroparasite. Note it down. The options are 1, Taenia solium, 2, Naegleria, 3, Acanth amoeba, 4, Trichinella spiralis. So out of this there is one worm which is not a neuroparasite, and that is Trichinella spiralis. It is found in the striated muscles. The question number 23. Plasmids are extrachromosomal self-replicating DNA molecules, all kinds of DNA molecules, all kinds of RNA molecules, all of the above. A very simple question, and the answer is the number 1, extrachromosomal self-replicating DNA molecules. Question number 24. Periplasmic flagella are present in 1, Helicobacter, 2, Agrobacterium, 3, Treponema, and 4, Chlamydia. The correct one is the option 3, which is the Treponema. Treponema are the spirochetes, and periplasmic flagella are present in the periplasmic space in the outer membrane of the cell wall. The question number 25. The effective method of sterilizing heat sensitive liquids is the option 1 UV radiation, option 2 beta propiolactone, option 3 exposure to ozone, option 4 is filtration. So out of this, the correct one is the filtration. In the filtration is carried under vacuum and the membranes are used. These membranes of various pore sizes. There can be the 0.25 micrometer pore size or there can be the 0.4 micrometer pore size. The question number 26. Animal disease that can be transmitted to human beings are called option 1 epizootiology, option 2 zoonotic, option 3 enzootic, option 4 is panzootic. So out of this, the correct one is the zoonotic diseases. The next question number 27. Fermentation of glucose molecules gives the following number of ATP molecules. The options are 4, 40, 2, 38. I hope you have guessed it. Yes, the correct answer is the option number 3. It is the 2 ATP molecules. Which of the following immunoglobulin is first to be synthesized in a host immune system with response to a pathogen? The options are IgM, IgA, IgE, IgG. The correct is the IgM. The IgM is the first antibody to appear in an immune response. Question 47. Immediate hypersensitivity usually involves 1 mast cells, 2 IgG, 3 platelets, 4 all the above. So here the answer which is correct is the mast cells. Question number 48. Opsonins includes 1 perforins, 2 meganins, 3 C9, 4 C3b. The correct one is the option number 4 C3b. Opsonins are those molecules which aids the attachment of the foreign particle with a phagocytic cell, and C3b is a component of the complement system. Question number 49 says molecules having antigenic determinants but not exhibiting immunogenicity are adjuvant, hapten, mitogen or the super antigen. So here the correct option is the haptens. Haptens, these are those low molecular weight antigens which are antigenic but they are not immunogenic. So they are attached with the carrier molecule so that they can become immunogenic. Question number 50 says antigen binding sites. Remember, antigen binding sites on antibody molecules are called 1 epitope, 2 agritope, 3 paratope, 4 etiolope. The correct option is the paratope. These are present on the Fab subunits. These are the antigen binding subunit present in the Y-shaped antibody molecule. Question number 51 says a hybrid cell obtained by the fusion of a B lymphocyte and a myeloma cell can be selected to produce molecular antibody by using the medium containing 1 thymine hypoxanthine aminopterin, 2 uridine hypoxanthine aminopterin, 3 thymidine histidine aminopterin, 4 thymidine hypoxanthine aminopterin. So here the correct one is the option number 4. The medium which is used for the preparation of the monoclonal antibodies, it contains 3 components. They are popularly called HAT. HAT stands for hypoxanthine, A stands for aminopterin and T stands for thymidine. Question 52. Vaccination is the type of 1 natural acquired passive immunity 2. Artificial passive acquired immunity. 3. Artificial active acquired immunity. 4. Natural active acquired immunity. So I hope you have guessed the correct option. It is the option number 3. Artificial active acquired. Remember 3A. Triple A. Question number 53. Which of the following is a cell-mediated response? Option 1, type 1 hypersensitivity. Option 2, type 2 hypersensitivity. Option 3, type 3 hypersensitivity. Option 4 is the type 4 hypersensitivity. So here the correct one is the type 4 hypersensitivity, also called delayed type of hypersensitivity, in which a subset of T cells called the DTH cells, they are involved. So these were some of the questions which were asked in the entrance exam of MSc Microbiology conducted by Gurukul Kangri. So hope you will do well in the exam. Best wishes. Bye. Take care.